Now to our first presenter. Dr. John Halfman is an instructor at the Finger Lakes Institute and the Department of at Hobart William Smith College and the Department of Geoscience and Environmental Studies. He's been testing and sampling the water from Owasco Lake since 2005. A couple of his projects that he works on are the pollutants coming from non-point sources and water quality variables between the watersheds of our Finger Lakes. We anticipate his 2020 report from his efforts and research from Owasco Lake. Take it away, John.
Thanks, Rick. I'm going to try to figure out how to share my screen here. There it is, share screen. Go up to that one, share. So hopefully people can see this. I'll start the slideshow. Start the slideshow.
I can see it. Thank you.
Okay, thanks a lot. All right, so seems like every year I get to come and talk to you guys about the status of the water quality in Owasco Lake. Well, here I am again to look at the status of the water quality in Owasco Lake. I'll add, I'm a professor here in the environmental studies program. I also contribute to the Department of Geoscience. I'm a research scientist at the Finger Lakes Institute, and I get into a lot of trouble at Hobart and William Smith colleges. But anyway, there's a nice view of Owasco Lake taken by a small drone showing that we can have beautiful days that are ice-free and sunny and warm, unlike today. And as far as I'm concerned, hopefully it's going to warm up here pretty soon because I'm supposed to be out in the field here pretty soon. But anyway, I'd like to start with this slide. Back in 2005, some students of mine and I went to 8 of the eastern Finger Lakes, actually 7 of the eastern Finger Lakes, and looked at all sorts of water quality parameters and then ranked these lakes from best to worst. Some of the best ones were Skaneateles and Canandaigua and Keuka College, but way over on the right-hand side, the one that was worse was Owasco Lake. And I started showing this to people like Senator Nozzolio when he was still in office, and he got all excited, and people around Owasco Lake got all excited by this. And it precipitated some grant money to continue doing water quality work in the lake. And the first couple of years of doing some of this water quality work, here's Owasco Lake. I had a bunch of sites going from one end of the lake all the way down to the other end of the lake. So here's all the open water sites measuring total phosphorus, 20 micrograms per liter is up here, parts per million. Notice that there's a bunch of sites way down here at the extreme southern end where there's plume from the Owasco Inlet comes into the lake that were really high in phosphorus, suggesting that runoff of non-point sources is definitely impacting the lake. The variability down here is reflective of you sample where there's no plume, you sample where there's a plume, you sample where there's no plume, et cetera, et cetera. So that got me some more money to try to do something about this in 2007, and none of those southern sites had high values. I remember looking at this data and starting to shit some bricks and wonder what was going on, because I made a big point of this in the previous year. It turns out that the difference between the 2 years is there was a lot of rain in 2006, not a whole lot of rain in 2007. Without the rain, you never had the runoff events to then measure them while you're out in the lake. So all these sites down at the southern end looked like pretty much the rest of the lake that you saw in 2006. So what does extra nutrients do to a lake ecosystem? Well, I like to show a simple nutrient cycle where you bring in some nutrients, plankton, take them up, it feeds everything else up in the water column. When plankton and fish and everything else die, that dead organic matter is decomposed by bacteria, releasing the nutrients again. So you then end up with this nutrient cycle. The more nutrients you can bring in, aquatic ecosystems are really, really good about recycling these nutrients and keeping them in play for years to come. So the more nutrients you can bring in, then all these boxes tend to get a little bit bigger, as shown in this slide. So there's more dissolved nutrients, more plankton, more fish and other things. You end up ending up with some algal scum like cyanobacteria, maybe more nearshore weeds, macrophytes, whatever you want to call them. maybe zebra and quagga mussels come in. These nutrient sources could be from a variety of sources, whether they're from fertilizers or CAFO operations or wastewater, municipal wastewater systems or on-site septic systems, or just the erosion of soil and organic matter in the soil. All these things can come in, contribute to the dissolved nutrient load, making more plankton, making everything look a little bit more, than, in layman's term, yucky. Oops. So 2020, busy year, made even busier because COVID issues prevented me from hiring any students. So I had to do all their work plus my work. And it was a busy year, but you can divide it up into two main parts. I did some lake and stream monitoring. We'll get into the details of both of these here a little bit. That lake and stream monitoring is supported every year by Cuyahoga County. Apparently I'm a line item in the county budget every year, which, you know, that's pretty impressive, I think. And then the Everson Foundation provided me money to put my water quality monitoring buoy, go back, which is shown here. I've got to be careful with how close the mouse gets to things. They also gave me some money to put in some dockside sensor arrays to then investigate blue-green algae and HABs events in a little bit more detail. So to continue all this, probably best to start with rain. Rain tends to be a major contributor of nutrients into the lake. This graph here shows the amount of rainfall by season. So there's spring, summer, fall, and there's the mean for the year, going from 2011 all the way up until this year. You'll notice this year is way below normal, about 56% of normal if you look at the average value here, especially in the fall. Unbelievably dry. And if you believe some of the previous talks that I've been giving, more rain brings more nutrients into a watershed. Well, less rain should bring less nutrients. And as more rain brings more nutrients and degrades water quality, then there's a chance that less nutrients might allow the water quality to improve some. So let's look at some of the data and see if this actually works. Before I get into some of the data, I'm showing you Owasco lake levels. Here's last year. You might have noticed that lake level shown here in blue is below what the rule curve is. Don't fault the operators at the end of the lake. They just didn't have any water coming into the lake to keep. So lake level continued to decline. A similar situation happened back in 2016, where it was dry enough that the water level just continued down. Some years we have way too much water and they try to let a lot of it out through the outlet, and so on and so forth. It also impacted groundwater depths. Groundwater depths were extremely low last year compared to previous years, so we're also low in 2016. So everything tends to dry up, and as things dry up, there's less water coming into the watershed, bringing nutrients along with them, and things should, water quality should improve in the lake a little bit. So let's start with stream monitoring in the beginning. There's a number of sites that I have in Dutch Hollow Brook watershed, of another site down here that samples the inflow from the Owasco Inlet, and then a couple other sites here along the western arm of the lake, the western edge of the lake, sampling some of these smaller tributaries coming into the side to get that representation. Do stream discharge, do a lot of on-site work, and then do lab analyses for nutrients, total phosphorus, soluble reactive phosphorus, nitrates, total suspended solids. What I couldn't get to last year due to the lack of help is I usually analyze for dissolved silica, but I just couldn't get that done. So if you look at stream discharge, in summary, look at this graph down here in the lower right. Here's last year's average for Dutch Hollow Brook. Here's last year's average for the Owasco Inlet. Notice it's significantly less than some of the previous years. In both these cases, Owasco Inlet has more water than Dutch Hollow because it's significantly bigger watershed than Dutch Hollow. But if you compare it to previous years in each stream, a lot less flow. So last year was really dry and corresponding to the lack of rainfall that we had. Nutrient concentrations, about the same, maybe a little bit less. Here's the lake over here. Here are all the sites in Dutch Hollow Brook going from downstream all the way to upstream. Here's the Owasco Inlet at Moravia, and here's the two side creeks coming in the western side of the lake. If you look at dissolved phosphorus, some of these smaller tributaries in Dutch Hollow Brook have higher dissolved phosphorus concentrations. Both of them drain agricultural areas, whereas some of these others are relatively low in the lake. Note that these small tributaries on the western side are relatively high as well. That drain up in the upper reaches some agricultural activity in the far western side of what's going on in the Owasco watershed. If you look at fluxes, fluxes are nothing more than the concentration times discharge. Fluxes last year were unbelievably low. You can barely see the fluxes in these small tributaries because their discharges were so low. Look at fluxes along the on Dutch Hollow Brook. They're basically the same, suggesting that you have a steady input of material coming in along that stream. Moravia has much more material coming in because it's a much bigger stream. Its discharge is a lot bigger. It also has 2 wastewater treatment plants shown in this graph at the bottom. But the loading from the wastewater treatment plants has been under control since about 2009 or so. Both of them under DEC mandate to make sure that they take phosphorus out of the effluent, doing some tertiary techniques to keep things relatively clean for the watershed. This is great. It's really easy to control point sources like these. The DEC says fix it, and then the responsible party has to comply if they want to continue to discharge stuff into natural waterways. I also do some daily samples at Dutch Hollow Brook at this street right here, which is 38A. It highlights flooding during rain events versus base flow conditions during non-rain events. You can see the variability in stream height down here in red. Whenever it rains in blue, stream goes high, and then it recedes back down. It rains, it goes up high again, then recedes back down. It rains, goes high again, so on and so forth. Here we have the 2020 graph. Notice all these spikes, they're missing. It's because we didn't have rain. There wasn't a lot coming down, except early on in the season. Might have been a little bit of snow melt going on as well. We had a couple of events. If you look at the material coming down Dutch Hollow Brook, here's total phosphorus flux, and here's the total suspended solids flux. These rain events bring the majority of the material. What I mean by majority is almost always 95, 96, 97, sometimes 99% of all the material coming down the stream is because of rain events. Rain hits the landscape, it then erodes particles from the landscape and does some stream bank erosion and other things. All this is carried by the stream and it gets carried into the lake. If you don't know what a daily sampler looks like, here's one. Here it is right here. Notice it's got 24 bottles, so you can collect a bunch of different daily samples. There's a little programmer on top, which then directs the water to the right bottle. It's all battery powered. There's a tube from all this, goes back, tube from all this that goes into the stream that then collects it relatively automatically. So I have to come by once a week, sometimes once every 2 weeks, save the water, put in new bottles and let it go for another couple of weeks. Notice down here, the total load, both total suspended solids and total phosphorus was significantly less compared to some previous years, like 2019, 2017, 15, 14. The ones highlighted in red were relatively dry and had similar loads, very low loads as 2020. So less nutrients getting into the lake. That's cool. Here we have a summary of phosphorus coming in and going out in the lake. All the inputs and all the outputs as best I can summarize it. And if you look at this graph, it looks like from 2016 onward, things are relatively good in that the inputs seem to be balancing the outputs. Occasionally there's more going out than coming in, which is even better. Occasionally it's the other way. Actually last year, 2020, it was the other way because even though very little was coming in the lake compared to some of these others, what was going out was even smaller because the flow going out the lake was really small as well. We just didn't have water in the lake to then maintain flow going out the lake to carry some of the nutrients that normally get carried on any other given year. But I'd like to point out that the fact that these are low, this is when we started doing some remediation practices within the watershed, specifically geared towards non-point source materials. And I like to think that these low values are due to some of that effort as well. People always like to know where all these nutrients are coming from. Here's my best guess for phosphorus coming into the lake. You have municipal wastewater treatment plants. That number is well known because it's published by the EPA. You can get an estimate what comes in by shoreline septic systems. And then the rest of this, primarily in orange or yellow, that's what comes in from the tributaries. Here you have Owasco Inlet, there you have Dutch Hollow Brook, and then all the other tributaries like the ones I sample on the western edge of the lake are shown here. Notice that the majority of the material coming into the lake is from tributary input. That would mean non-point sources. Very little is coming in from point sources way down here. So if you really want to clean up the lake, the thing you need to do is control these non-point sources, and then you have a chance of trying to clean it up. 2020, last year, way over here, a little bit less coming in than all these other years. This is good. The ones that have high loads are probably are the wet years that we see, especially down here in 2011. I also get questions about all the geese that habitat the lake. This is on the same scale what 1,000 geese would do if they were there 365 days a year. That's the best guess estimate you can. Apparently people go around and follow geese and measure their poop and the amount of phosphorus in their poop. And there you go. That's where it would be on this scale here. They're not a major player to the total phosphorus budget of this system. So how are you going to curtail this degradation? Well, you want to curtail some of these runoff events. If you curtail the runoff events, then somehow hold the sediment back from these runoff events. Maybe to do that, you control roadside ditches a little bit, maybe control drain tiles a little bit. I'm pleased to see that there's a drain tile talk here in a little bit after mine. Maybe we want to have stricter controls for lakeshore septics, but I'm pretty confident what the Department of Health and the county's doing is pretty good. But if you wanted stricter controls, you could connect all the lakeshore residents to municipal wastewater treatment facilities. That would lower the load a little bit. Down for agricultural things, the more BMPs you can put in, the better off you are. Maybe you put in buffer strips. Maybe you have more responsible manure spreading. Maybe you have you remove the pee from the animal waste before you spread it. All these ideas have been incorporated into the revised rules and regulations. Hopefully they'll get passed here soon. They're all going to get incorporated into the 9E plan, which Dr. Walters is going to talk about here a little bit. All these would have a pretty good impact at reducing nutrients coming into the lake and making things cleaner. Now, did the lake actually clean up? Well, I have 2 sites in the lake. I have a water quality monitoring buoy in between. Do all sorts of analyses at these sites, including nutrient concentrations, CTD casts, Secudus depths, plankton tows, a bunch of things. All of it's done from my pontoon boat, as you can see down here. It's a fun thing to drive around with. What I didn't quite get to again was dissolved silica because I didn't have students. It's debatable whether we can hire students this summer coming up as well. So hopefully I can, my guess is probably no. But anyway, Secudus depths. Secudus is this little disc here. You lower it down through the water column until you can't see it. You measure that water depth. And then I have annual values in each one of these blue bars. There's 50% of the data. There's the smallest depth. And there's the deepest depth down here. The scale is backwards because clearer water is down towards the bottom. Notice that 2020 was basically the same as some earlier years. 2016, things got worse. But basically, we've been in this middle category here, which isn't oligotrophic, isn't eutrophic. Oligotrophic being really clean water, eutrophic being really productive water. We're somewhere in between. We're a mesotrophic system. Superimposed on here is some diamonds. This is from the CSLAP volunteers, what they've measured, and it's great to see what they're doing is very similar to what I'm doing. Look at phosphorus and chlorophyll concentrations. Phosphorus did go down in the lake, as the lack of rainfall did predict. But on the other side of the coin, chlorophyll concentrations went up. So this is relatively perplexing until I figured out that last year there was a greater abundance of cyanobacteria in the offshore sites. They float at the surface, so they're going to influence my surface grab, and my surface grab was here. So there was more algae at the surface where I sample it, which then led to a higher concentration just because there was more blue-green algae out there. So as usual, this lake will vary between mesotrophic and oligotrophic conditions. More indicators were oligotrophic than mesotrophic, so the lake water quality improved a little bit, as the lower rainfall should have predicted. This is good. Looking at the plankton that's there, here I have fluoroprobe data, which measures between diatoms, green algae, cyanobacteria, and cryptophytes in the different colors. Probably the most important color to know is the black. That's the cyanobacteria. This is 2017, all the visits out on the lake. Here's 18, there's 2019, and there's 2020. There's a disturbing trend here in that you don't see any black bars in these 2 years. You see a few more black bars in 2019 and a lot more in 2020. It suggests that we're getting more and more cyanobacteria out into the open water, to the point where some of the plankton toes in gray and in black, that's the cyanobacteria, they dominated. They were the majority of the algae that's out there based on count. This is based on the concentration of the chlorophyll that's in the organisms. This is a little bit disturbing, but it could be explained by last year was a little bit windier than previous years. Maybe the wind brought more blue-green algae out into the open water from the nearshore events. Time will tell. Next year's data will be pretty interesting to look at. As far as cyanobacteria goes, here's all the different blooms that were recorded. This is based off the DEC website. Every time they put a new one in, I put it on my graph. The dominant wind direction, according to the buoy, was from the southeast, something along those lines. So it suggests that more calm conditions happened along the eastern shore, allowing for more blooms along the eastern shore than over here on the western shore. Maybe that slight shift in wind direction, usually the wind's directly out of the south or maybe out here of the south-southwest. That slight shift in wind direction perhaps was responsible for significantly more blooms based on the Bloom Watch volunteers looking for blooms around the lake than any other year on record. Maybe more people were looking, maybe they're better at finding these things, I don't know. It might be due to the shift in winds. That's what I'd like to think anyway. That's what the data is trying to tell me. If you're interested in why there's still cyanobacteria here, well, there's a couple of things that I can point out. One is there's always been cyanobacteria in the plankton. From day one, back in 2005, so here is annual concentrations for Honeoye Lake, Canandaigua Lake, Keuka, Seneca, Cayuga, Owasco, Skaneateles, and Owasco. So here we have Owasco Lake, the lake that you guys are concerned about. There's always been some cyanobacteria in the plankton. It's only recently, in the past 4 or 5 or 6 years, that they decided to have nearshore blooms, as shown in this picture that's sort of covered up by everything else. In case you don't know, here's a plankton net. You just drag it through the water. Water goes through the net, but the mesh size is small enough, plankton stays trapped. It's congregated down in this little cup. You pour it into a sample vial, you bring it back to the lab, and you start counting algae. I don't count algae. I hate biology. My wife counts algae. She likes them. So that works really, really well. So to try to figure out why we have these blooms in these lakes, I've been trying to look at some of the environmental preferences for blue-green algae or cyanobacteria. One of them is they like warmer water. So my data, I've collected water temperatures, so we'll look at that. They like sunny skies and calm conditions, so we can see if they're there as well. They like excess nutrients. And here I think is the biggest bugaboo for the Finger Lakes region, because most of the Finger Lakes are nutrient poor. So we'll investigate that a little bit more detail as well. So as far as warmer water, here's surface water temperatures. Behind here is Owasco Lake. All the times that I went out. In blue is when I started putting out the buoy, so there's a lot more data incorporated in that data set. I have 25 years of surface water temperatures from Seneca Lake shown here. Notice that both of these, if you just put a best fit line through all the data points, we've had warmer water in more recent times. And what's even more disturbing is the warmest years, like in Seneca Lake, the last 5 years, warmest, that's when they had their cyanobacteria blooms around the shore. So warmer water suggests more cyanobacteria blooms. For Owasco Lake, it started around 2012, started there, graphs a little off. Some of these are a little bit cool, but the rest of these are nice warm temperatures. Interesting. This warming, by the way, is probably a global warming signal. Every year, the lake seems to be getting about 0.2 of a degree centigrade warmer each year, which, from year to year doesn't seem like much, but over the long term is relatively significant. So there's one condition: warmer water seems to be working out just fine. We are getting warmer. This picture by drone suggests that Owasco Lake can get very sunny, can get very calm. Now, the unfortunate thing here is there are more sunny and calm days than blooms. In other words, what I'm trying to say here is you don't get a bloom on every sunny and calm day. So there's something else going on besides just it has to be sunny and calm. That something else is probably nutrients. Now, I mentioned that the Finger Lakes are relatively nutrient poor. Well, if you look at the mean annual concentration of cyanobacteria, and then convert that concentration of cyanobacteria using Redfield ratios to the mean total phosphorus concentration, there's 20. And if you compare 20 to the phosphorus that's available in the open water sites, rarely is it above 20. There's 20. Some of these blooms have significantly more than 20, about an order of magnitude, perhaps more. I'm suggesting that the open water probably is not the source of nutrients for making these nearshore blooms. Something else can happen. So then you spend 3 years breaking propellers, getting into the nearshore zone, trying to measure concentrations there. Notice that here's offshore waters. There's a bunch of nearshore sites. All these nearshore sites basically have exactly the same concentration as the open water. So it's not the water column that has the nutrients to support these blue-green algae blooms. It's got to be something else. So then you get a little bit more sophisticated. You get a number of dock sites. I have a bunch around Owasco Lake and also a bunch more around Seneca Lake, where you measure the weather. You put a camera, an automatic camera, which then takes a picture of the lake so you can document blooms and when they occurred and how long they occurred. You then put a deployment tube here where you can put in a water quality sonde that measures dissolved oxygen and algal concentrations, temperature, and the salinity of the water. You measure temperature as well in a number of different aspects. I had one down here at Fire Lane 20, at the end of Fire Lane 20, a bunch of other sites up here on the eastern side. Oops, go back. Whoop, that's going the wrong way. Up here on the eastern side, because that's where they get more blooms. One at Burgess Point, a couple at Martin Point up here. We were hoping also to look at the sediments and measure the amount of sediment organics, the macrophyte densities, the zebra mussel surveys as well. We were hoping to get some drone surveys. This is what I couldn't do because of the lack of student power, but I got just about everything else done. If you look at what the camera showed, they can show blue-green algae blooms or cyanobacteria blooms really, really nicely. It's either that or the water's clear or it's turbid because it's windy. You can actually see the drift of some of these cyanobacteria blooms because of longshore currents. You can measure the number of blooms that happened at all these sites. It went from 0 at one site to 9 or 10 in 2 different years to 17 to 12 or 14, so it's variable. I like to say that cyanobacteria are really fickle. The duration of these blooms can last anywhere from about 20 minutes all the way up to one bloom lasted 9 hours at one site. The mean here is anywhere from 2 to 3 hours. So just because you don't see it doesn't mean it won't happen later in the day, or it didn't happen earlier in the day. It turns out that the owl volunteers at these sites saw about 1/10 the number of blooms that I detected with the camera. So, you know, it happens, but they're only out once a week, so that's not too bad. The time of day that blooms like to prefer is from noon on, but I've seen blooms as early as 7 a.m. all the way up to 7 p.m. So there can be blooms just about any time you want. I also got to see a bunch of ducks and kayaks and other things in some of the cameras. Be careful when you're swimming next to any of these things. Probably some of the most important data that I got was from these weather stations is looking at both wind direction and wind speed. First, if you compare buoy wind speeds to the nearshore wind speeds, the nearshore sites have significantly lower wind speeds. This is primarily due to shoreline configuration. If the shoreline accepts it, if the shoreline is downwind to the predominant wind direction, then you'll get a lot of wind. If it's not, then the shoreline will block that wind. The other thing that's different is the dominant wind direction at some of these sites. It shifts from predominantly southerly, which is the same as the open water, to westerly, to one site had northerly winds and not very strong northerly winds. So it suggests that the shoreline location and the shoreline configuration can change the dominant wind direction. This to me means that the decrease in wind speed promotes the chance of having more blue-green algae blooms in the nearshore region. These altered directions means one site could have a bloom because it's calm, whereas the other opposite shore site might be too windy to have a bloom and won't have a bloom. So that starts to increase the variability in bloom events around the shore. It then promotes the fact that I call them fickle. They might not like that if they heard me, so hopefully they aren't hearing me because they might get back at me and have lots of blooms next year, but we'll see what's going on. It did definitely document the fact that not every good, calm, and sunny day had a bloom. So there's got to be more going on than just the meteorological conditions. That more that's going on has to be somehow related to nutrients. It did show that people say that blue-green algae blooms happen in the warmest water. Well, here it's warm, there's no bloom. Actually took a dip in water temperatures to then get a bloom, another big dip in water temperatures to then have those blooms. The same, this was back in 2017. The same thing happened to a smaller extent here in 2020. The dips will happen because there's a wind event right here. That wind then mixes colder bottom water up to the surface. That wind event can also disturb the nearshore sediments and perhaps release nutrients trapped in the sediments into the water column, which then might support the bloom on the next calm and sunny day. Some evidence from some of these water quality sondes that were put at the docks around this site. There's one site in particular where macrophytes, these nearshore weeds, accumulated inside the deployment pipe. Once they started to accumulate inside the deployment pipe, it happened right here on that day, the oxygen within that pipe started to go down to 0, started to become anoxic. It means to me that there was significant respiration going on of those microphytes. With that respiration will then release lots of nutrients. So you can see the respiration of the macrophytes as the chlorophyll concentration goes way down, and it takes about a month for all that organic matter within the tube to then decay away. What's even more startling was as soon as you start releasing some of those nutrients, the blue-green algae start to really take off, but only inside that pipe. They didn't migrate outside the pipe. I didn't see them in the cameras. It only happened within that pipe. So if you can get decaying weeds, then you have a very good chance of getting blue-green algae. It then promotes enough nutrients for the blue-green algae and cyanobacteria to take off and bloom. Looking along the shore, there was a shoreline just south of the Burtis Point site. had a bunch of rotting weeds along one shoreline. You go to the next neighbor, that next neighbor did a really good job about raking up all these decaying weeds. Where there was weeds, HABs. Where there was no weeds, no HABs. So it suggests maybe a good remediation would be to rake up these decaying weeds. It then suggests that if the wind is blowing in a certain direction, the downwind accumulation of all these weeds will then be a site of a blue green algae bloom, a cyanobacteria bloom. Another simple experiment that I did was to take mud samples and put them in flasks, add filtered water, lake water, so that had no algae in them, and then just sit them in the sunshine for a couple of days. Within a week, they all had blue-green algae blooms, suggesting that there's sufficient resting cells. Blue-green algae, cyanobacteria make a resting cell that settles into the mud, to waiting for the next time to then have a fortunate bloom. And there's sufficient nutrients in those muds to then support cyanobacteria blooms. So for remediation, probably the best thing to do is try to remove all that organic matter that's dead, that washes up along the shoreline. Because if you can do that, then maybe it's going to reduce the amount of cyanobacteria that gets there. More importantly, let's continue to work really hard to reduce nutrient loading to the lake, because the ultimate source of all these nutrients is what comes in from the streams and what's coming in from these non-point sources that contribute to the stream and supply of the stream. There's a final thought that I like to put out. This is a depressing thought. So if you don't want depressing news, turn your cameras off or turn your sound off. But get back to the simple nutrient cycle. Dissolved nutrients feed plankton. They feed everything else in the ecosystem, including fish that people like to fish for. When these things die, bacteria decompose them, release nutrients so the plankton can take them up again. I have a bunch of pictures of different plankton. There's a zooplankton that eats them. The fish eat zooplankton, or you need little fish to eat the zooplankton so the bigger fish can eat them. I don't know, it's an interesting, relatively simple system. That's why I like aquatic systems, they're relatively simple. Then we had zebra mussels and quagga mussels come in. They're a lot better at taking up plankton than perhaps going to fish and other organisms. So you then start with this new pathway. There's plenty of zebra and quagga mussels in Owasco Lake, so we're probably having this pathway. So that means there's less nutrients going into fish. That's a bummer, because I know a lot of people within the lake like to go fishing. This has really impacted Seneca Lake, in my opinion, and the fishing success rates have gone way down in Seneca Lake. But here's the depressing thing. The depressing thing is once you get a lot of cyanobacteria, they're a lot better at taking up these big pulses of nutrients than perhaps anybody else over here. So they can go right into HAB events. You get nice blooms, as you see in these pictures on either side. Then when the bloom dies, it goes into the bud. Bacteria can decompose them to release more nutrients to then make more HABs. So once you get these nutrients into these systems, you then end up with a new nutrient cycle, which means you get HABs, you will continue to have HABs, and you're always going to have HABs unless something can be done with the nutrients that are sitting around in the nearshore mud. These zebra and quagga mussels have probably promoted more nutrients into the nearshore zone from something called the nearshore shunt. And now the cyanobacteria are taking over and probably doing just fine. What's disturbing to me is you get a good bloom like this. Once it dies, it goes right into the nearshore mud. It gets decomposed, releasing nutrients for the next event, some few weeks later. So that's the depressing news that I have. If you didn't like it, hopefully you didn't hear it, but that's perhaps the way we're going to go for a few more years, if not decades. With that, I'd like to stop. Hopefully that was around 30 minutes because that was the time limit that I was given. I'm willing to answer a few questions now. Probably can't get to all of them, but if people do have other questions, feel free to send me an email. My email's up here in the upper left-hand side. I also have some of the reports, some of the annual reports that I've been writing about this, about Owasco Lake, the water quality in Owasco Lake, and also my research about cyanobacteria. That's available at this website. You have to scroll down to the bottom of that page You got to go through graduation ceremonies for some of my kids and so on and so forth. Them riding horses and other things. But way down at the bottom of that page, there's links to all these reports. Feel free to download them, distribute them, do anything you want with them, print them out, then use them for firewood. I don't care, whatever. But there's a lot of information there that you might you might want to look at. So with that, I will stop this share so other people can take over. And I will try to stop my presentation. There we go.
Thank you, John. Rick here. I have several questions. We'll try to get to as many as possible. Again, John said, go to his website. If you have other questions, you can direct them to us at allo.org and we'll try to get the answer. So one of the first one is John, monitoring water quality, how many days per year do the significant rain events occur? 10 days, 20 days?
Depends what you mean by significant, but there's usually rain about once a week. Now, the really big events that might happen 2-3 times a year, or at least when I'm out there measuring. I'm not out there in the winter months or the early spring months when there can be more significant rain or snowfall events, as you guys well know.
During dry periods and low lake level, does this affect the internal cycle loading? And if so, less cycling from the bottom sediments, more or no change?
Good question. My first feeling is because you have lower lake levels, then the wind and waves can then impact deeper water depths to then get some of the stored nutrients that are in the mud in slightly deeper water depths. So I think it's going to have a bigger loading, and it might have contributed to the greater number of blooms that we saw around the lake last year in Owasco Lake.
Okay, is there a correlation between nutrient loads and macrophyte growth? Can Dutch Hollow inputs or internal lake cycling and legacy nutrients account for lush growth?
I haven't measured macrophyte growth well enough to be able to answer that.
Okay, curious how the load from the shoreline septic system is estimated.
Based on studies that have done in other lakes, they actually, the only nutrient source around some of those other lakes is lakeshore septics, and they actually figured out the increase in nutrients that way.
John, can you use volunteers in place of paid students?
Yes and no. It's always nice to be able to train them and have them work for me all summer long.
Well, they'll contact you if that's a possibility, right? How's that? The lake has been overtaken by seaweed growth, which was told to be from phosphates and nitrogen coming from the inlet and the south end. Would you confirm this? And from these phosphates and nitrogen came the seaweed, then the zebra mussels came in, consuming the algae, blocking the sun. Seaweed growth then grew dramatically. What are we doing about this?
The answer is yes. Nutrients coming in from all streams, not just the Owasco Inlet, are contributing to the growth of weeds and whatnot. What's being done about it is, for example, in the Owasco Inlet, they've made detention basins, which will then accept the flow, and during events, to then that flow gets into these big basins and all the suspended sediments settle out before the clarified water continues on down the lake. That's one way to try to control these things. Another way to try to control these things is to put in buffer strips around farmers' fields, so that as the runoff hits the edge of the field, then most of the suspended sediment will then drop out. The suspended sediment is key here because it has the nutrients attached to them. So if you can drop out those suspended sediments, that's great. You can put in catch basins as well, grass waterways within a field if there's significant gullies within the farmer's field. There's a bunch of things you can do. They're starting to do it. The Soil and Water Group here in Cayuga County have been doing a great job trying to get grants to help farmers to do some of this stuff. I'm really impressed, and I think some of the data that I'm showing is showing the fruits of some of their efforts.
I can add, John, that Al has been involved with the Soil and Water Conservation District on a ditch remediation program. In the past 2 years, over 20 miles of ditches have been hydroseeded, and up to about a mile have had rock lining to prevent erosion. But there are many more miles of ditches and streams that need to be taken care of. So there are many things underway. Again, like I said before, much has been done, but much more is needed.
I agree.
Dock monitoring station, how deep is the lake water there in that? I think at probably Martin.
So it's about a meter deep.
You talked about the HAVs hotspots and the wind and weather direction related downwind accumulation. Do your camera sites observe you around?
No, I put them up or somewhere in mid-July and take them down somewhere after the end of October or during October, somewhere in there. They wouldn't last year round.
Can there be community interest in not only removing weeds, but adding living shoreline protection to minimize erosion and take up nutrients with planting? I say, yes, there is interest.
I would hope there'd be interest in just cleaning up the weeds that wash ashore. That'd probably be a major thing. Just put them in the back of your yard in a compost pile.
There are online sources with those, how to be lakeshore friendly living. And so
Yep.
Contact us and I'll and you if you want further reference. Should we place seaweed mats around the lake too for these?
You can. Keep in mind that those same macrophyte beds are good rookeries for little fish, which then grow up into bigger fish that which you like to go out fishing for. So getting rid of all the weeds, maybe not a really good idea. I mean, they're an essential part of the ecosystem to have some weeds.
Okay, a couple more questions. My girlfriend and my female dog both had multiple UTI tract infections last summer with no visible HABs in the water. How can we as residents know it is safe to be in the water? You know, the health department's guidelines are if you see a HAB, don't go in the water.
That's right, and that's what I always say. If the water looks green, don't go in, because you don't know if that bloom is secreting toxins or not secreting toxins, and you can't do it without a complicated test. That's really expensive.
It's
the little bit of data that I've seen is if there's no bloom there that's visible, then there's no toxins in the water. So it should have been not due to cyanobacteria. Maybe due to something else.
I'd like to add one more question that was emailed earlier. The municipal water intakes and the treatment that they're doing are the carbon systems that they've installed in Auburn and Owasco for their treating their water to remove cyanobacteria and toxins. Are they a reliable long-term method for that removal?
Yes.
Okay. Do you? I know they've been effective and they were relatively quickly installed compared to other options. The next question is, supposedly cyanobacteria do not exist below the thermocline, that layer deep in the lake, correct? And if so, would extending the intakes deeper prevent cyanobacteria as a long-term solution to not?
Okay, so most photosynthetic organisms don't like to be below the thermocline, because below the thermocline there's no light, and they have no way to really survive because they can't photosynthesize anything. It doesn't mean that they aren't down there. It's just that they congregate themselves up near the surface of the lake because that's where there's more sunlight, so they can do more photosynthesis.
The estimate I heard from Auburn, they think they may have, if they wanted to do that, they'd have to go at least 100 feet deep, and that would be $10 million plus if it's even feasible. So, and you're saying they may still be drawing in water that has cyanobacteria. They went that.
Yep.
So, okay, I think that's we need to move on. And so, any questions, please email them to John and us. And thank you very much, John, for your presentation.
Oh, you're welcome. It's always a